Okay, good afternoon, everyone. And in today's class, we will continue with where we left. We were discussing about cell signaling pathways that are um, involved in stem cells as well as in cancer stem cells, right? So there are many signaling pathways that influence, but we were discussing about three of them: Notch signaling, Wnt signaling, and hedgehog signaling. So the first one was Notch signaling. In this, we discussed about Notch that is produced as a single peptide. Then it is processed. There are some steps of addition of oligosaccharides, and then there is first S1 cleavage, that is by furin. A single polypeptide is made into two. So now you can see that three domains in total. One is the extracellular domain, the intracellular domain, and the transmembrane domains. So when it binds to notch ligand like Jagger, delta, and so on, the notch ligand, there will be conformational changes, and then S2 cleavage will occur, which is mediated by ADAM protease, and that cleaves off the extracellular domain. So what is left is the transmembrane domain and the NICD, notch intracellular domain, which is monoubiquitinated, and then eventually it forms into an endocytosis. I mean, through endocytosis, it is internalized into the cytoplasm, and then S3 cleavage occurs by gamma secretase. And this notch signal, NICD, right, will be translocated into the nucleus, where it will act on target genes involved in growth and cell division and so on. Coming to Wnt signaling, we discussed about the Wnt is the signal, WNT, and then it binds to a receptor called as frizzled, and frizzled will bind to DVL, and that because of that binding, beta catenin gets stabilized or increased in concentration within the cytoplasm, which otherwise would have been degraded, as in this case. And beta catenin will enter into the nucleus, and there it will decrease the production genes that are involved in adhesion and increase the genes that are involved in migration. So if just for revision, if Wnt is not present, the one that is present here, it's not present, then frizzled is not active and it does not activate DVL. In that case, the beta catenin is acted upon by GSK 3 and axin, APC, and so on, because of which beta TRCP will ubiquitinate beta catenin, which is as a result of polyubiquitination targeted for proteasomal degradation. So because of which the genes here in the nucleus that are involved in migration are not as expressed, right? So in today's class, we have to discuss about hedgehog signaling. And as I told you before, there are many protein factors, and so many things that we don't have to mug up. Just know the three ways, two things: the signal, you should know; the receptor, you should know; the doer, you should know; and then the genes that are activated. That's the consequence, right? This is what signal is coming, what is receiving it, what is triggering the activity, or whose activity is going to act upon the genes. Okay. So in hedgehog signaling, it is there can be three different types of signals. The one of it is called as sonic hedgehog, or Indian hedgehog. I'm actually ignorant about why, on what basis they may name it. And then there is desert hedgehog. So these are usually the mammalian ligands for a protein. These are the three different types of signals in this case. And then this is patched is the receptor that is, along with the other interactions, interact with the ligand. Patched normally has a function. It has another interaction with a protein called smooth end. This smooth end is inhibited by patch, right? If there is interaction between the hedgehog and the patched, then it inhibits smooth end, because of which the a protein called as gli protein has three different kinds of domains which are referred to as 1, 2, and 3. That is gli, and it can be processed in two ways. Either gli can be as 1 and 2, or it can be as 3. So in this case, when sumo is not inhibited, it is binding to several other proteins, and then it will process the gli. Gli is, because of multiple interactions, it is processed in such a way that this the domain 3 is degraded. So what is left is 1 and 2, gli 1 and 2. Gli 1 and 2 is now active, and it goes into the nucleus, and will activate transcription of several genes that are involved in cell cycle regulation. If there is no ligand, or the hedgehog ligand is not bound, then patched is not in an active state. So because of which, sorry, I said something else wrong here. Okay, my mistake. So normally what would happen if the ligand is not present? Smooth end is maintained as a vesicle, and it stays there, and patched will thus inhibit um, gli 1, 2, 3 is processed differently. Gli 1, 2, 3 will be targeted and processed differently because of beta TRCP by ubiquitination. What happens is gli 1 and 2 subunits, these two subunits get degraded, and what is left is gli 3R. Okay, that also goes into nucleus, but here it acts as a repressor, gli 3 repressor, and it represses the target genes. Let me take another try with hedgehog. 1 second, sorry. So normally what would happen, let's see, without and then with, I should have started it that way. Yes. So when there is no ligand, patched is active, and it will inhibit smooth end, because of which gli is processed differently, and gli 1, 2, 3 1 and 2 gets degraded, and what is left is gli 3, which acts as a receptor and after entering into the nucleus. In the presence of ligands, the ligands are hedgehog. The name of the signaling pathway is based on the ligand. It binds to patched, because of which the inhibition is no longer there. So smooth end, the vesicle binds to the cell membrane, and it's on the cell surface now, and because of which gli is processed differently. The part 3 is degraded, and what is left is gli 1 and 2, which is in an activator, and it goes into the nucleus, and it will activate genes that are involved in cell cycle. Okay. Yes. So we have learned these three different cell signaling pathways, and the idea is to know how we can probably interfere. We are learning this is still as a part of cancer stem cells. So we would want to know if there is a way to specifically inhibit these particular signaling pathways so that we can control the cancer stem cells. So the question is, we have 4 places, and signal, receptor, doer, and then the genes. And which one is likely the best place? Every place is important. One thing we would want to also know is a signal can activate a receptor, and receptor can activate multiple doers, right? Or there can be other steps also, and then that can activate one gene multiple times, right? There is a signal amplification, they call it. Majority of the signaling pathways you will have signal amplification. So just one molecule, and then the consequences could be drastic because you have some amplification here. So thinking on those lines, which is a better state to influence? If there is no signal, if you're going to attack or somehow inhibit the signal, that is a good thing. Lack of signal can be done, or error, or what do you say, degradation of the signal, or blocking the receptor is also one of the strategies to do. And then having inhibitors for the doer is also one another strategy. Let's see a few of them. So this is a diagram illustrating the same ones, and then they are providing some compounds. We don't have to mug up those things, but you can still think of those, which are these target sites that can be there. So here, this is for notch signaling, right? Notch signaling, we can have soluble receptor decoys, and that is a way to somehow be able to interfere in the interactions between receptors and the ligands, delta or jagged, or you can call it as notch ligand also. We can try monoclonal antibodies. There are many things that can actually influence or interfere in the interactions. We are more interested on this side of it. So we can try to add uh, gamma secretase inhibitors. I hope you remember that gamma secretase is involved in S3 cleavage, right? That will generate the production of NICD, notch intracellular domain, which acts as it is the doer in this case. So if there are several compounds people have been testing that that could be used to interfere in the activity of gamma secretase. Similarly, there are also people are also trying to do other, because there are accessory proteins which will bind to the NICD, along with which the transcriptional activation of the target genes happens, trying to interfere by using MAM L1 inhibitors. There are also some other ways of trying to do, because the target genes are produced. I didn't mention that. If there are genes are produced, how can we stop transcription is one way. How can we neutralize the produced mRNA, or somehow try to degrade the mRNA that is being produced? Those are some of the strategies. So here, small interfering RNA or microRNA, you can siRNA technology, you call that small interfering RNA, which are, say, if this is the mRNA, right, 5 prime and 3 prime, we would also call it as sense RNA, sense mRNA, because it has the start codon and stop codon. So this small interfering RNAs, microRNAs, and microRNAs work by somehow producing small stretches of antisense RNA. So there are several proteins, dicer and other things which would recognize them and will break the mRNA into pieces. Okay, so that is trying to interfere at the level of the genes that are being produced. Okay, so you don't have to mug up, but when we think about what to design uh, the drugs against, we can always find out the target genes. If we know specific details, then you can do it in silico to start with, if we have the protein sequence, or the protein sequence and the three-dimensional structure of the, say for example, gamma secretase, if we have the structure of it, then we can probably design novel compounds and that will interfere in the active site of gamma secretase, which will decrease the affinity for the notch or may interfere with the interaction itself or the activity of proteolytic activity itself. Okay, but let's finish these things. So in the case of the Wnt signaling, we can add antibodies against Wnt, monoclonal antibodies. I hope you know how the monoclonal antibodies are made. There is HAT medium, HAT medium, hypoxanthine, aminopterin, and thymidine. And fuse two different cells, which is one is antibody producing cell, the other one is, say, myeloma or some cell that has continuous growth capacity. You can fuse them and grow them. I mean, there is a whole genetic way, a screen that is made so that, and then you raise them again in HAT medium. Only those fused cells which can grow continuously and produce antibodies will survive, and the rest will not. And now one cell will produce antibodies against one epitope. And all those such antibodies will then be cloned as monoclonal antibodies. Say from one cell you're taking, then that antibody is specific to one epitope. What is an epitope? Epitope is a part of an antigen which is recognized by an antibody. So monoclonal antibodies can be raised. What people do is usually say, I will just cook up. Maybe they take rabbit or so little large mammal, and then they inject Wnt. If it is the human Wnt, okay, which is assumed that it is different from that of the rabbit one, and the rabbit might be producing antibodies against Wnt. Say, for example, this is Wnt, okay, and it has three different antigen epitopes. The whole of it could be an antigen, and this region is one epitope. This region is another epitope, and this one is another epitope. So the antibodies raised against it could be against this epitope. I'll call it as epitope A, epitope B, and epitope C, right? So all these antibodies together will be referred as polyclonal. Each of them is coming from a different one. But if we could isolate a cell that is producing anti-epitope A and then perform the hybridization, cellular hybridization, and then produce, then we will get monoclonal antibodies. And those can be then used, injected into the body, and they were supposed to bind to the Wnt. If they bind to Wnt, it is unlikely that they will bind to the respective receptor. So that is how we can minimize the amount of Wnt that is available to the cell. We can also try small molecule inhibitors like I described before. We can also try antibodies against frizzled antibodies if they assume that the receptor is something like this, which is antibody is already binding, then Wnt may not have probability of interaction with this, and therefore it will not be active. That is one another way of things. Wnt is bound by two proteins, LRP and frizzled. So we can also try to use LRP inhibitors. And this is a compound for against all. When you see a mark like this, that means it is inhibition. Okay. So we can try many of these compounds. There are several compounds that are being tested. I don't think something unless there are many of them might be in clinical trials. Okay. Similarly, for um, hedgehog signaling. There is robot nicotine. Um, some of them are smoothened antagonist against smoothened so that it doesn't function. And there are some other compounds like this. Okay. As I said, we don't have to really mug up these things. Okay. So what we have learned is the signaling pathway. In during examinations, it's very likely that we might ask any one or two of these, and how you try to control the signaling pathways. So you should try to know if one or two inhibitors. The important things, first you should know the cell signaling itself, the pathway itself, and then how it can be interfered. Okay. So we have discussed many things, and I just want to go through a few of those that are supposedly on. One second. The sorry, the stem cell niche a little bit, and then we can stop. Which is already we have discussed, and we need to just. It is about melanoma stem cell, and what is the genetic hit is where is the mutation happening. Okay. So melanoma stem cell, this is melanoma is, I'm not saying melanocyte stem cell, or melanoma is already meaning that it is the cancer stem cell. Okay. And then we have transit amplifying melanoma cells, and then we have differentiated, which will give rise to differentiated melanoma cells, which are increasing in mutations, and so because of which their diversity is changing or increasing diversity. We don't know exactly which of these may have contributed to the formation of the melanoma stem cell. It could be any of them, differentiated melanocyte or a melanoblast or transit amplifying cells or the melanocyte stem cell. I would place, if I have to pick only one, and I see that there is largest probability, which doesn't mean the others are impossible. The largest probability is with transit amplifying cells. I'm saying probability. I'm not saying that he's the answer for this question. Okay. But yeah, in different cancers, it could be different as well. Some people also think it's because of cell fusion. We have a cell and it fuses with another cell, which will form tetraploid, which will then, upon divisions, may become or become close to diploid. During these things, during these events, 4n and becoming 2n, and so there could be chromosomal rearrangements, and because of which some of the cells may form or gain the capacity of stemness and tumorigenicity, and that is nothing but the formation of the cancer stem cell or melanoma stem cell in this case, right? So what does the stem cell niche has? It has heterologous cell types. We have a this is cancer stem cell or even adult stem cell too. It is almost the same. We have several different types of cells. That is the heterologous cell types. There are several extracellular matrix, and there are several glycoproteins, matrix glycoproteins, and there are signals coming from external side which might be having a role on how this stem cell behaves. It's true for almost the whole cell and the whole body itself. This is how every cell is. You don't find any cells, maybe the liquid cells in the liquid tissues, maybe they are like kind of independent, but otherwise majority whatever we are discussing here is true for all the cells, all the cells in our body, majority of them. And what may also influence is the local metabolic conditions. Usually the stem cells or cancer stem cells, or at least the adult stem cells, are in low metabolic condition. They don't, compared to the surroundings. Sorry. So if we can, the idea of trying to discuss this is, we will, when we discuss about a therapeutic strategies in the next class, we will talk about it again. If we can, this there is a cancer stem cell, and it has these three-dimensional interactions, right? With physical interactions, mechanical forces, the chemicals, everything included. And if there is a way that this cancer stem cell or stem cell in general is moved out of its niche is something where it is located. If it moves on outside, then it does not have the same interactions. And because of those interactions, if lack of those interactions, it might differentiate. So the one of the other ideas is, somehow how can we interfere with these interactions? If we can interfere with the interactions, then the cancer stem cell could become a differentiated cell, and then probably we can probably be able to treat or eliminate cancer stem cells. Okay. So these are some of the things that you might want to remember: CD133 and CD20 and ABCG. Then there are also not signaling. We discussed about not signaling, Wnt signaling, and sonic hedgehog signaling pathways, and a little bit about stem cell niche. Okay. Just 1 second. Sorry about it. I wanted to have some other discussion actually. Okay. So I wanted to also have an introduction to mammospheres and breast carcinoma. This is the third cancer stem cells that we will be discussing about. We will not, because yours is a modified syllabus or simplified, because from the course we used to call it as a stem cell biology. It's now fundamentals in stem cell biology, fundamentals of stem cell biology. We'll not have a chance to discuss all the signaling pathways here, but I would want to still talk about a few things. Breast cell cancer stem cells, because it's important. So breast is a mammary, it is mammary gland. It's important for lactation, and the parent will give has parental care as a baby. Majority in most of the species, you see that they are dependent on their parents. So the parents should provide food. In animals, some of the animals like snakes and other things, are those that of frogs, many of those lower, not lower, primitive in nature in a way, they are born ready to take on the environment and survive. But mammals and some other, say birds, for example, they do a lot of parental care, right? Their parental care is to fetch food and give it to the baby, chicks, right? That is their parental care. I think the reptiles onwards, majority of them, they are born ready to take on. So this strategy is to have as many as children as possible, and because of which those that have been able to survive, able to fetch food and eat, and they will grow stronger, they will survive. In birds and some other animals, they are dependent. They cannot fly, they cannot move as much, so they are dependent on the parent, and the parents will fetch food and bring it to the chick. That is part of parental care, right? So in mammals, what we will find is parental care has taken a new step. It is not going to fetch food, but directly provide the food to the child, and that is milk in the form of milk, right? The parent may get the food or not, but the milk is, or the food for the child is produced. One second, sorry, is produced, and the child will always have the food, at least until the early stages. As humans, we are much more dependent on the parental. I think probably ours in humans, you will have the longest duration of parental care. Maybe in monkeys and something like that, maybe it is 1 year. Chimps and so on, maybe 2 years. And the humans, in the earliest of the humans, probably 10 years. And now it is like 30 years or 20 years if it is in our countries. In Western countries, maybe 15 or so. So we live in joint families, you can count it as even 50 years also, just saying for the fun. So how did the humans or mammals in general come about this? How did it become evolutionarily this way? It is thought, I think we will have some slides about it, but the organization, you would see that there will be muscle here, and then there is a lot of adipose tissue, and then we have several lobules that is containing the mammary alveoli, and they produce the milk and they pour into the alveolar duct, and then we have the tip nipple. There will be milk production, right? The baby, and this lactation will only happen upon delivery, or we will see in the cycles. But before going, yeah, let's have just a look at it. How the evolution, the model for evolution of mammary gland might have happened. It's a model, okay? Xanthin oxidoreductase, lysozyme, some of these these enzymes are involved to provide protection from bacteria and bacteria especially. So that some of the glands may have, or the genes may have evolutionary developmental biology, you can think of it, may have invaginated, and with so larger surface area will have more production of um, xanthin oxidoreductase and lysozyme, and eventually these have evolved. There's in skin glands, they they are protective in nature, but then eventually developed into during evolutionary stages, okay, not like in human, this is not how it happens. Maybe in the most primitive mammals this might have happened, and slowly over the periods it has come about like this. And and then eventually they might have developed into lactating mammary glands. So when do we require these mammary glands to be? The mammary glands should be there when there is a small kid or baby to feed, right? Otherwise, the mother would be investing too much of resources, nutrition to produce milk, and the milk, if there is no baby, there is no point of producing the milk. So everything is hormonally hardwired to produce milk only during the time of nurturing a child, otherwise not. I want to take you a little bit outside, and there is a country called as Martinia, I think. You can also try it out. I can't remember exactly. I saw a documentary on National Geographic, and this child is being force fed. I mean, they're feeding her using force. What you see here, the feet are between two legs, and on this side, these two sticks will be tied together, and then the mother is holding the stick, pressing the feet in between. And this is her child, and she is feeding her camel milk or goat milk that is available, because it is in their culture that the child should look obese, and obese is a sign of confluence, or what you call it as, it's a kind of pride to be, and kind of sign of richness. So they force feed to make the child obese. And if you see the documentary properly, they would say, if the child is very thin, it is a disgrace, kind of disgrace to the family. And in some other countries, I think it's North African countries also, there are kind of something like hostels, where the girl child is left in the hostels, and in the hostels they would feed her continuously and constantly a large amount of food until she becomes very obese, right? We If you I want to compare just our own living itself, what are we in comparison to different other organisms that we see in the on the how are we different from them? We are eating breakfast, we are having a lunch, we are having maybe some snacks and then dinner and maybe midnight snack. Our our daily routine is filled with food. If we take any of the and how do we get this food? We get this food for almost free. That means free, I said, is about, do we spend energy to get food or not? We don't spend energy to get food here. We get it by, say, parents at least, or we would have earned money which might not have been really physically troublesome or physically hard work, right? It is maybe mental work. But you can think of my profession. I am right now sitting and teaching, right? There is not so much of physical work, mental work, yes. And I'm speaking, maybe that is the most effortful thing other than the thinking process, what to say, what not to say. I'm sitting and teaching, and then I get salary and money out of it, out of which I buy food and I'm eating, right? There is a serious disproportion between the food, amount of food I'm taking, and amount of physical activity I'm having. And that's going to be a problem for me. It is That is how these lifestyle diseases actually also become exaggerated. So I hope you're trying to fit your own lifestyle into it. While you are young, the metabolic rate is high, and because of which you will actually be healthy enough. But otherwise, as you keep aging, we need to somehow increase continuous or um, constant work during our daily routine itself. What would that be if it is if it is a earliest or ancient human? The ancient human would have to hunt for food and then eat the food, right? He is spending so much energy just to hunt for food. And there might be days where he or she might have gone hungry or starved for it. Our daily life is filled with so much of food that we actually don't know what true hunger is unless you're fasting or other things. So that is one more thing we need to bring into perspective when we think of these things. And here we have a cultural activity where one is supposed to be obese, which is otherwise a disgrace to the family, or it's considered not taking proper parental care. We don't know how these cultural practices come about, but it could make some sense if we are trying to justify it. Okay, in desert areas or so, there might be several days where food is not available, in the ancient times at least. For several years together, several days together, maybe somebody may not be having food. sufficient food. And in that case, if somebody is obese, maybe they are likely to fare well because they have so much additional reserve food to burn before they might die out of hunger. Okay, that is a logical explanation we could probably give to the cultural practices that happen around the world. So, what happens during from birth to the lactation, and what is the cycle that happens? In a newborn, we might find these small branches starting to form, and then we have around a lymph node, and then around 4-week version, the lobules or these things will keep on growing, the branching, and then eventually the growth hormone estrogen and so on, they will induce the formation. They will grow to a certain age. Around the time they are reaching the puberty and maturity, so some other molecules or hormones like progesterone, prolactin, and other things will increase the size of the breast mammary glands. And then during pregnancy, because of prolactin and other things, they will grow into full size. And once they deliver, the lactation will start. In that lactation, they will give milk to the baby. And once the weaning is over, meaning that the child stops taking breast milk, then the grown breast will revert to something called as involution. It becomes smaller in size compared to how much it was during the lactation time, and it'll reach this stage. So this is a cycle that happens every pregnancy, conception, delivery, lactation, and then involution. Right? This is what keeps on happening. So, I mean, what I'm trying to also discuss is, or you will just see this, I will finish in one more slide maybe, and then we'll have a discussion. A resting gland is something like this, and during pregnancy, it grows in size, and during lactation, it will start producing a large amount of milk. Right? It's not like mammary glands are always producing milk. It's only happens during the time after the delivery. If you see the alveolar cell, it has so many vesicles, and each of these vesicles is filled with, these are the alveolar cells, and they have vesicles of food, the droplets, fat, and other things which are secreted into this small lobule like thing, and this will empty it into the ducts, and from the ducts it will reach to the baby. So all these milk droplets are produced as vesicles within the cell. Okay? And you can also see myoepithelial cell. Those may have the constriction capacity to press so that the milk is produced, sent out. This is how a lactiferous duct may look like in and around. And during lactation, you can see each of these cells filled with so many of these milk lipid droplets that are present. Right? Milk is a very interesting emulsion, isn't it? So, yeah, I will not go into the cancer and other stuff, but I would want to discuss the daily lifestyle and what should the cycle actually be. We have seen this, the previous one, where this is the development of the individual to adulthood or reproductively active or right. And then we have these cycles of conception, delivery, lactation, and back to stage. So typically, the humans should, if we consider a monkey or a chimpanzee, they do not have any plans like, I mean, not like careers and so on. And at least even the ancient humans didn't have those. But now we have careers for women, and that is very important for women to have, because our society right now is dependent on financial liberty. The more financial liberty, the more respect, and we are going to live by those things, and this is what we want. In the If you look at different organisms and the societies or the groups of organisms, their individuals didn't matter. Individuals didn't matter as long as the group is fine. That means some individuals were struggling and some individuals were having a lot of privileges. Okay, but now we have democracy. It's not just the right is not might is not the right. Even the weak can survive, and weak, I said, is about physical strength. Until humans and until the monkeys and many of the things, the brute force works. Of course, there are other skills required about how to manage other people, showing empathy when required, but physical dominance was a prerequisite to be the one with privileges. But in democracy, we have everybody has some privileges. Everybody survives, right? Everybody gets, at least they get his reproductive chances for reproduction. They can find somebody and find reproduce along with them. If we looked at animals, in chimps and other things, they don't have reproductive rights. Not everyone has the reproductive rights. Only the alpha male usually gets. Maybe there are betas. That means the second after alpha may get some rights, but otherwise majority of the males do not get, and that's why there is a lot of infighting. They fight constantly. But in democracy, we kind of assure everybody a chance and right to live and right to reproduce, as long as they can find a partner, and its finding is not so much. And that is the reason how the human civilization has prospered so much. So we are talking about the females, right? So females, we typically maybe around 12 years old or so, they undergo puberty, maturity. That means, biologically speaking, at least, they are ready for reproduction. That means they started um, producing eggs, and of course, it's very dangerous to the mother if she gets pregnant by before 18 or so, because the human head is huge, and during the delivery that causes a lot of problems. And the society construct is also is based on economic status, right? If you want to bring in a child and raise the child, you need to have so many other resources that the humans have invented, some things like money, and those are many things that will allow the growth. So the child, a mother of very young mother, may not be able to physically tolerate or bear the reproduction itself, the childbirth. And the second thing is economic needs required or resources required to supply or the take care of the child. So we can consider, but otherwise in the wild, if you are or in where civilization, what we call civilization, has not touched, people around this age may also start giving birth, and that is actually the biological nature. So evolution and biology are kind of ruthless. A 12-year-old cannot give rise to or cannot tolerate birth and dies, the evolution will let it die. There is nothing to do about it. Only those that will survive will survive. That is how every species, I think I started this whole course about how to keep the species healthy. And what evolution does, evolution is not a kind of an entity or something. That's a process. Whichever is strong will survive, the rest will not. Humans, we have invented many things because of which even the diseased people or disabled people or differently able people or many people can survive. We can treat, prolong, or postpone death, which might actually otherwise occur earlier. So now the schedule has changed for humans, right? We need to study, and then we need to get a job, and then partner, and then we get to something like what you call it as settled. That is, by the time they become a mother, they are at least 25 years old, right? So 12 years biological maturity for reproduction, and then we have 25. What would have happened in the wild? By 12, this girl may be having at least 6 pregnancies if it were in the ancient times. But now it's postponed to 25 years, and maybe the person will have 1 or 2 children, and then the rest of the life again no children. The question here is, what is the breast made up of? What is breast designed for? It is to for childcare, and it has inherently the capacity to grow and become again back to its original state. And this cycle should happen, and that is when it is the natural way of life for the breast. The first again, there is one more question I keep pondering on is, in which animal did we ever see? If you can find an answer to this question, please let me know. I have been trying to look at it and try to understand how this works. In which animal do we see breast so prominent? We see breasts in only 2 different types of organisms I know, or 2 types of organisms. One is humans, and the other one is the domesticated ones. Because again, we are selecting them for milk, right? Like goats or cows. If you look at, if we, to my understanding and knowledge, if we look at wild animals, the animals that live by themselves, they will only have prominent breasts that can be observable only during the time of lactation. In all other times, it's almost difficult to make it up. There must have been the evolutionary uh, selection for, or it could be a cultural practice that we in humans we may have selected for humans with enlarged breasts. Okay, so what I'm trying to imply is, if we have selected for breast or enlarged ones, then we might be selecting mutations that may be involved in breast enlargement. It is just a theory. So we can make these comparisons about how human evolution, how human structure is, and with that of wild animals. We should never do it with domesticated animals. In domesticated animals, some of the cows are so the interbreeding, through breeding they have come across made some cows that whose the mammary glands actually obstruct their walking also. It feels so sad for the animal that is having to carry those. But I would like you to check on this, and then try to discuss. We will discuss about breast cancer, and I hope I've given you enough to think about why there is so much of breast cancer incidence in human population. Okay, I'll stop here. If you have any questions, please ask. Any questions?